Due to the sporadic nature of somatic cancers, the number of detected variants is growing exponentially. This presents a challenge for labs and clinicians like you to rapidly and efficiently analyze and interpret an increasing number of genetic tests with the latest scientific evidence, drug approvals, and treatment guidelines. Users have expressed that the ability to promptly deliver NGS test results to oncologists is crucial for improved patient outcomes, and yet they find themselves spending hours annotating somatic variants. In this video, we'll show you how you can empower your somatic bioinformatics pipeline with just one comprehensive database called HSMD, helping you streamline the variant annotation process and confidently recommend biomarker-matched therapies in a fraction of the time. But first, what is HSMD? HSMD, or the Human Somatic Mutation Database, provides comprehensive information on human somatic mutations. The database is regularly updated, providing you with the most up-to-date information on variants, associated treatment options, off-label therapies, resistance markers, and regional or disease-specific clinical trials. HSMD includes data from our manually curated knowledge base, as well as real-world clinical oncology cases analyzed and interpreted by QIAGEN's Professional Clinical Interpretation Service, formerly N of 1, making HSMD a trusted resource for any clinical lab. With HSMD, you can rapidly drill down to variant-level information. Let's take a look. Here, you can easily search for your variant of interest and open up the variant information page. On this page, HSMD provides a description of each variant, including the functional impact with hyperlinks to supporting literature sources, external links to ClinVar, dbSNP, and OncoKB for further validation, complete alteration details, including the position of the variant, the alteration type, as well as the functional impact with a descriptive comment and hyperlinks to the supporting literature, actionability tier, and population frequency as well. You can access the up-to-date literature for your variant of interest in the bibliography section, and sort the literature for relevant functional evidence, for example, by simply clicking on Category and selecting your literature type of interest. In the drugs section, you can find approved drugs and off-label therapies for your variant. And the clinical trials section allows you to identify both regional clinical trials and disease-specific clinical trials. So, using HSMD, you can easily report this variant as a Tier 1 activating missense variant associated with 2 drug indications and 10 clinical trials, for example. This highlights how HSMD's centralized, up-to-date variant information can help you reduce time spent on annotating and reporting clinical variants. Thank you so much for joining us today.